I think the most interesting aspect of my research is that the decision making methods that we develop are so generally applicable to so many different kinds of systems. And the reason I believe that's the case is because we follow this really tightly coupled approach of first rigorously modeling the system behavior or dynamics using principles of basic maths and physics, then using those developed models to do stochastic planning and control, and then actually generating system response and studying that data, and based on the analysis of the data, refining the models that we started off in the first place to understand system behavior. The potential impact of my research to the society as large is tremendous. I'll give you a couple of examples. So one is related to the work that I do at the micro scale. So we use a specific type of equipment known as optical tweezers, which uses a single source of infrared low power laser, but then we use a computer controllable hologram to split that laser into potentially hundreds of different beams, and we are able to control each of these beams independently in 3 dimensions. And with each of those split beams, we can actually grasp, just like a robot can grasp an object, and manipulate an object if it is of few microns in diameter. Now, the place where this tool is most useful is for manipulating biological objects such as cells. And in this context, one of the most exciting applications that I recently started on with our collaborator in the Department of Bioengineering is on how to better develop engineered heart tissues. So people have been trying to do this for several decades now using techniques like bioprinting and micromolding, but they have encountered difficulties when they want to print really small scale objects which are few microns or tens of microns in diameters. Now, using our optical tweezers, we are able to precisely grab and manipulate those endothelial cells or stromal cells, for examples, which can then be precisely positioned on top of growing cardio muscle cells to actually create vascular networks in vitro before the tissue is actually grafted into the host animal body. And we believe that if we can demonstrate that, it will tremendously improve the reliability and efficiency at which we can create these engineered tissues. Moving on to a totally different length scale, another project that we started working on is related to more efficient supply chain management for large scale assembly manufacturing operations, and we are primarily looking at aerospace industry in this particular context. So the problem with aerospace assembly manufacturing is that when the assemblers want to get parts, they need to get them from hundreds of globally distributed suppliers, and these parts have lead times of the order of 200 to up to 700 days. So when the buyers place these orders, the suppliers commit to their best possible guesses on when they can deliver their parts, but the whole world and the economy of the world can change in the matter of 2 years' time. So their estimates did not be necessarily accurate, and they keep revising their estimates over course of time. But nevertheless, it turns out that the actual delivery dates for these parts never seem to tally that well with the estimates that the suppliers provide, which causes a lot of problem to the buyers in terms of maintaining their inventory levels as well as scheduling their assembly operations efficiently. So we are developing a fairly sophisticated stochastic model to make those estimates much more accurate and robust, using lots of historical data and not just relying on the supplier provided estimates. So that can make manufacturing much more competitive, and it gels really well with the initiative that the US government is now trying to push for. I think the most interesting aspect of my research is the fact that the decision making methods that we develop are so broadly and generally applicable to so many different kinds of systems that operate under uncertainty. And the reason I believe that's the case is because we follow such a tightly coupled approach of first rigorously modeling the system behavior or dynamics using principles from maths and physics, then using those developed models to do stochastic planning and control of these systems, and then learning from the system behavior, which is basically data driven analytics, so that we can refine the models that we started off with in the first place. I believe that the potential impact of my research to the society at large is tremendous, and I'll give you a couple of different examples from two very different laying scale applications. So the work that I'm doing, for example, with microscale systems, there we are using a specific type of equipment known as optical tweezers, which has been a popular tool at the hands of biophysicists, but right now we are trying to automate operations of these tweezers so that it becomes more useful to the broader engineering community as a whole too. So what it does, it uses an infrared laser source, and we can split that single laser beam using a computer controllable hologram in real time to generate up to hundreds of different laser beams. And we can control each of these split beams independently in 3 dimensions, and then it kind of acts like a micro robot. So just like a robot, you can use it to grasp objects. We can use this optically actuated micro robot to grasp and move around tiny objects at the micro and nano scales. And the specific cool application that we recently started working on with our collaborator in bioengineering is on how to develop better engineered heart tissues. So people have been trying to do it for quite some time now using techniques like micromolding and bioprinting, but they usually run into difficulties when they have to print really small scale objects like individual cells or groups of cells that are few microns or tens of microns in their dimensions. Here is where optical tweezers play a vital role. So we can use the laser traps to move around and then precisely position and pattern individual endothelial cells or stromal cells on top of growing cardiomyocytes, which are the muscle cells that form the key ingredient of our heart. And that way we can sort of hierarchically assemble the entire heart structure artificially in vitro before we ever need to graft that tissue inside the animal body. And we believe that if we can do this successfully, we would come up with a much more efficient and reliable method to create better performing heart tissues. Another example of where my work is very impactful from a very different length scale standpoint is our recent work on using predictive modeling for supply chain management. And here we are looking specifically on aerospace assembly manufacturing industry. The issue with that industry is that no one really builds all the parts in-house. Instead, they get all these parts from many globally distributed suppliers, and typically these parts have lead times of the order of 200 to 700 days. Now the whole world economy may change dramatically in the period of 2 years. So when you place the orders, the suppliers may give some estimates of when they expect to deliver the parts, but those estimates keep getting revised constantly, and their own capacities and capabilities may change over time. So it's very hard for the buyers to really know when the parts are actually going to show up. That is why we are building a fairly sophisticated stochastic model which can learn from historical data using many other attributes and not just the supplier provided estimates. And that way the buyers can better manage their inventory levels because they would know more accurately when the parts are going to show up, and they can also schedule their assembly operations much better so that they can meet the end customer demands better. I can talk a little bit about another of my projects which also I think can have a huge impact on the society, that is on understanding the effectiveness of different treatment options for breast cancer patients. And in this we are collaborating with Dartmouth Hitchcock Medical Center, which provides the only comprehensive cancer care facility in rural New England. So we have data over a period of 3 to 5 years for a large number of female breast cancer patients who were diagnosed and treated in their facility. and the thing that we are doing is basically aggregating data from multiple different sources. So for a lot of healthcare informatics, people tend to look only at one source of data, and as a result they sometimes miss the correlations between data attributes or factors that you may otherwise get if you, for example, connect genomics data to the medical administration records to the tumor data. So that's what we are trying to do using a novel technique called associative memory that kind of mimics how our own human memory stores and accumulates data. And using this associative memory, we are able to identify interesting correlations between factors like patient's age and the effectiveness of certain types of chemotherapy agents, for example. So that way, for breast cancer patients, while a treatment is going on, we can, or the physicians can make some course corrections, because we all know that chemotherapy has lots of adverse side effects. So initially the physicians may prescribe a certain dosage or level of drugs based on what they think is most needed to cure the patient, but based on how the patient is actually responding, whether that patient is recovering well or she is developing some other forms of side effects, there is the possibility now, using our technology, to change the level or the duration of these reagents so that the long-term impact to patients' health gets improved.